Question 1. Lipids are important biological chemicals that play many different roles in cells and organisms. And here's one type of lipid. Name the type of lipid. And 2 uses. So type of lipid. This is a triglyceride. And uses. And in organisms, so we can go for anything at all. Things like protection of organs. So when you do a heart dissection, it's got lots of fat around it. Thermal insulation. So a layer of fat in your skin. Electrical insulation. For example, myelin sheath around neurons. All sorts of other stuff. You could put metabolic, water, buoyancy, helping animals float, waterproofing, for example, waxy cuticles, things like that. How you would test. So this is how you would test. So you've done this, and 2 marks. So it's going to be saying do what the test is and what the result is. So for example, mix your sample with ethanol, then water, and then that will it'll go cloudy, or you could possibly say milky, or you could say a white emulsion. So pretty straightforward so far, just remembering stuff, no real tests so far. Describe how components Y and Z differ from component X. So if we are looking at Y and Z, we appear to have this area here that isn't present in X. So X is saturated, but Y and Z are unsaturated. So that means that X has got as many hydrogens as possible for the carbon, whereas Y and Z, they haven't got as many hydrogens, and they've got a carbon-to-carbon double bond. So Y is okay. That's yeah. You could say Y and Z have carbon-to-carbon double bonds, and so on. Okay. So we've got a description there. And then actually the other 2 marks are for the second part, which is why it is recommended that humans eat a higher proportion of lipids containing Y and Z. And these are, eat more unsaturated fats, and you might have heard of polyunsaturated fats, because these decrease levels of LDL, which is low density lipoprotein. So these decrease your levels of LDLs, or you could say the opposite, saturated, increase them. And then this reduces the risk of heart disease, or you might have another technical word like atherosclerosis, atheroma, or so on. The science behind that is actually disputed, to be honest, but that's what the textbooks are currently saying. Then we've got a different type of lipid, and looking at it, we've got our fatty acid tails there, we've got our glycerol remnants in the middle, and then we've got something with P in, so phosphorus, or phosphate head there. When these lipid molecules are extracted from the plasma membrane of cells, they will arrange themselves as shown. And that's our phosphate head. These are our fatty acid tails. Phosphate head is hydrophilic, and fatty acid tails are hydrophobic. So that's the information that you know. Why they arrange themselves that way? Well, the phosphate heads are attracted to the water, so phosphate heads attracted to the water, and the other way around for the fatty acid tails. So fatty acid tails are repelled from water, and we can associate these words that we've used here. So hydrophilic, so hydrophilic, and fatty acid tails are hydrophobic. So those are the two marks there. And then we've got a little bit of a calculation, a little bit of a teaser here, where the plasma membrane of a cell has a surface area of this number, 140 micrometers squared. What would be the area of water covered? The calculation is this 140 divided by 2, times by 2 is 280. The reason for that is because normally these would have a bilayer. So if we're showing them like that there, well, these would have a bilayer in a normal cell situation, and so they'll be twice as big an area if you're not in this double layer, this bilayer. So we just need to put that into words there. So phospholipids are arranged in a bilayer in the cell membrane, but a single layer in water. And we've got a strange little experiment here where we are shining UV light onto a genetically engineered membrane, which these proteins are fluorescent. So we shine UV light, and then that's after shining, then it gradually returns back. There's a number of different experiments that do this very same thing with dyes and all sorts of things. But basically, we're doing something to the proteins, and then suddenly they appear to spread out and replace themselves. And that's what we need to explain. So it says, explain how these images provide evidence for the currently accepted model of a plasma membrane. So let's say what this model is. The model is the fluid Mosaic model. They're actually going to give us a mark there. And we need to mention what's happening to these proteins. So the proteins can move within the membrane. So that's the fluid part, and the mosaic refers to the fact that these proteins are embedded in the phospholipids, so it looks a bit like a mosaic. And then explain what's going on. So the fluorescence, so the fluorescence is actually the area around here, and then the laser beam, they lose their ability to fluoresce. So fluorescence, spell that if you can, fluorescence is restored to the laser beamed area after 5 minutes. So this idea of proteins that can fluoresce, diffusing or moving around in this layer, and after 5 minutes it appears quite normal. So 15 marks.